I'd like to introduce our first speaker, Eleni Mimitou. Eleni is a senior research scientist at the New York Genome Center. She pursued a doctoral degree at Columbia University and a postdoctoral training at the Memorial Sloan Kettering Cancer Center in the field of genome integrity. As a member of the Technology Innovation Lab here at the New York Genome Center, her research is focused on single-cell genomics, with emphasis on tools for obtaining multiple modalities of information from high-throughput single-cell assays. With that, I'll hand it over to you, Eleni.
Okay, let me share my screen. Okay, you can hear me and you can see the presentation?
Yes.
Okay, great. Hi, everyone. Thank you for tuning in. I'm very excited to launch this seminar series by presenting our preprint on scalable multimodal profiling of chromatin accessibility and protein levels in single cells. All right. As we all keep witnessing, and just what Rahul just said, combining modalities in single cell assays is increasing in popularity and utility. While mRNA is usually the common currency, several methods have been developed to simultaneously profile gene expression with chromatin accessibility, perturbations, spatial information, just to name a few. In the Technology Innovation Lab, we have been interested in integrating protein detection with single-cell RNA-seq workflows. And we find proteins attractive because they can be diagnostic of cell types and phenotypes, but also importantly, due to their high copy number, they make an easily captured high coverage modality that could complement the usually sparse single-cell data. And this way, we can enable more robust cell type discrimination. In the past, with CiteSeq and ExciteSeq, to bridge this gap between gene expression and proteomic analysis, we attached an oligo to an antibody that detects a protein. This oligo has some unique features, so it brings an antibody-specific barcode, which functions as an antibody-specific identifier, and a capture sequence that allows it to get barcoded in parallel with mRNAs. So it could be either poly-A-tailed in 3' technologies, as we've done with CiteSeq, or it could be the reverse complement of a TSO, the template switch oligo, as it's done in the 5' VTJ kit in the context of ExciteSeq. In this preprint, we try to bridge epigenomic and proteomic analysis with ASAP-seq. ASAP stands for ATAC, with select antigen profiling by sequencing. And this work was done in collaboration with many groups, including Aviv and Life at Broad, Vijay and Caleb at Harvard, and Shimon and Kelvin at Osaka University. A very brief background on how Ataxic works and what it helps us measure. Ataxic stands for Assay of Transposase Accessible Chromatin by Sequencing. And it takes advantage of the fact that TN5 transposase inserts only in regions of open chromatin, which is traditionally considered to be active transcriptionally. TN5 importantly brings along adapter sequences that get inserted, and those serve as PCR handles. So upon sequencing and alignment of these fragments, we can see them piling up in accessible regions that either are in between nucleosomes or span nucleosomes, and they help us identify the so-called attack peaks. Currently, single-cell attack is enabled by different technologies. For this, either combinatorial, microfluidic, nanowell, and droplet-based. For this work, we used the 10x droplet-based system, which is using nuclei as input. The nuclei are incubated with transposase in bulk, and subsequently they're partitioned into droplets alongside the barcoded oligos. Upon release, these barcoded oligos interact with the augmented DNA fragments via the TN5 adapter sequence and produce those barcoded single-stranded DNA fragments. But to make protein profile worthwhile, we needed to maintain the connection between nuclear content for attack and cell surface marker information. So we had to use whole cells as input. Coincidentally, Leif and Caleb, as Deus Ex Machina, developed a mitochondrial single cell Ataxic in an effort to retain and profile mitochondrial DNA mutations in parallel with chromatin accessibility. And this method uses whole fixed cells as input in the 10X platform. So we reasoned if we label these cells with our oligo conjugated antibodies, we could potentially combine protein detection with Ataxic, giving rise to ASAP-seq. To test how this cell preparation befits cell surface marker detection, Kelvin here performed a flow experiment to test whether there's any protein signal left after the cell permeabilization steps. Antibody staining was assessed during different steps of cell processing, starting with stained, unfixed, fixed cells, fixed and permeabilized, and finally fixed, permeabilized, and incubated for 1 hour at 37 to mimic the TN5 transposition step. As you can see, every step takes a slight hit in the signal detected, some more than others, but at least for the 3 markers we tested here, the signal is clearly well above background. And encouraged by this, we decided to try the sequencing-based detection and use oligo-conjugated antibodies. Now, instead of making a new class of antibody conjugates, we wanted to find a way to employ the already available, optimized and extensive repertoire that BioLegend has produced in their Total 6 series. As you can see, there's currently available more than 1,000 antibodies available. These are tailored for single-cell RNA-seq platforms. So to repurpose them, we took advantage of the fact that the barcoding reaction in droplets is a linear amplification reaction. So we designed a bridge oligo that binds to the existing protein tag and serves as a template to allow the tag to extend and acquire sequences, shown here in gray, that are necessary to interact with the barcoded oligo present in droplets and get barcoded in parallel with attack fragments. Now, by modifying this side of the bridge oligo, the one that anneals the protein tag, we can bridge any Total 6 product in a very economical way. In one of the experiments that we performed to benchmark ASAP-seq, we used PBMCs that were stained with a panel of 9 major blood markers, the ABMK panel. After staining the cells and washing away unbound antibodies, the cells were permeabilized according to empty single-cell Ataxic and subjected to transposition, followed by barcoding in droplets with the bridge oligo spiked in. The recovered cells clustered in attack space, yielding expected populations. And importantly, we could reliably detect canonical cell protein markers in these cell types. And the patterns mostly, if not always, correlated with gene activity scores. If we take CD3, for example, it stained the T cell cluster, which is mutually exclusive for CD4 and CD8. And CD11C, which stains the monocyte cluster, and it's mutually exclusive for 14 and 16. For further technical benchmarking, we wanted to test simultaneous the staining and capture of two different BioLegend families, Total 6A and Total 6B. So we stained in 1-to-1 ratio the same antibody panel. And we spiked in now 2 bridge oligos during the barcoding reactions. We observed that detection is essentially equivalent. And moreover, we recognized that the mitochondrial DNA retention is not always desirable. So we tested the lysis condition. It's more similar to the one used in the 10X protocol. And while this condition depleted the attack libraries for mitochondrial DNA fragments, the protein detection was largely unaffected. So mitochondrial DNA capture in ASAP-seq is optional, and users can decide whether to use this modality or not. Moreover, because ASAP-seq employs existing reagents, it's readily available with, it's readily compatible, with hashing. Hashing is the antibody-based cell labeling that allows sample multiplexing and overloading by means of reading the barcode attached to the antibody that used to pre-stain cells before cooling. In this experiment, PBMCs were split in 4 aliquots and stained with 4 Total 6A hashing antibodies. And following sequencing of the ATAC and the tag libraries, we recovered about 14,000 cells, which is more than the maximum loading recommended by 10x. And those were successfully demultiplexed in 4 singlets and 6 doublet clusters. Furthermore, we took advantage of the fact that the ASAP-seq workflow includes a fixation and a lysis step, so it lends itself to test intracellular staining. For this experiment, cells were first stained with the ABMK panel, the 9 extracellular markers, and after permeabilization with 3 intracellular antibodies against ZAP70, granzyme B, and perforin. The recovered cells were clustered in attack space, yielding, again, expected cell types. And here I'm zooming in the T cell NK cluster and highlighting counts for 3 extracellular markers on top and the 3 intracellular on the bottom. These staining patterns agree with known expression patterns within T and NK cells, where ZAP70 stains both CD4 and CD8 cells, whereas granzyme B and perforin are specific for CD8 cytotoxic cells and NK cells. This specific staining is indicative to us of a reliable intracellular staining, and we are excited to explore this further with more intracellular markers. Once we knew the assay works well, and with BioLegend's help, we expanded the number of protein markers we looked at. In this example, we applied ASAP-seq to human bone marrow mononuclear cells stained with a total-seq antibody of 240 markers. We permeabilized the cells following the empty single-cell Ataxic to maintain mitochondrial DNA, and simultaneously interrogated accessible chromatin, mitochondrial DNA mutations, and antibody tag abundances. About 11,000 high-quality cells were identified, and they clustered in 21 distinct groups, spanning the major hematopoietic lineages. And they displayed concordant protein marker decoration, spanning from progenitors to myeloid to lymphoid cells. Since the mitochondrial DNA retention protocol was used, we also detected 99 heteroplasmic mutations. Most were distributed across the different lineages, but there were some, I think, 2 that were interestingly depleted in the erythroid lineage. Importantly, we looked at protein expression in the course of monocyte differentiation. Here I'm showing only 61 out of the 240 markers that were used over the pseudo-time trajectory. And I want to emphasize the fact that this is a very useful catalog, and this process has been traditionally characterized with a limited set of markers, but ASAP-seq is not limited in the markers that can be interrogated, measured, and thus can provide deeper profiling of the cell marker diversity. Also, given that ASAP-seq and employs existing reagents, as I mentioned, that are used in single-cell RNA-seq workflows, we were placed in a unique situation where both ASAP and site can be performed in the same stained cell sample. To test this, we used a model of T-cell activation. A PBMC sample was split into 2 aliquots, one stimulated with a tetrameric anti-CD3, CD28, while the other was cultured in the absence of stimulation and served as the control sample. After 16 hours, we stained with a total CK antibody of 227 antibodies. And post-staining, we withdrew a portion of the cells, the native cells, to run site-seq, while the remaining cells were permeabilized for ASAP-seq. Similarly to what we observed with FACS, there's a slight decrease in UMI counts as measured by ASAP-seq compared to site-seq, which is not surprising considering that the permeabilization steps disrupt the cell membrane integrity. But when we compare antibody signal change upon stimulation captured by the 2 methods, we see that there is high concordance across the panel. That tells us that ASAP-seq performs as well as site-seq, despite that slight drop in UMIs detected. It was reassuring to see that substantially upregulated are canonical T cell markers like CD69 and 25. Oh, sorry. And on the other hand, there is a noticeable reduction in CD3, CD28, and TCR alpha beta, presumably due to the receptor internalization upon the stimulation. Also, the fact that each sample was co-labeled with the exact same antibody reagent makes it possible to integrate data from these two assays together. And here in this analysis, Caleb integrated and harmonized ATAC and RNA with protein data. On the left plot, we focus on T cells, and we show fold change on in the protein level y-axis, transcription x-axis, and chromatin accessibility by color scale. This kind of analysis can help us identify changes that are differentially detected across the modalities. Let's take, for example, CD3 and CD69, which is also shown in the UMAPs on the right, are the upregulation of 69 and the downregulation of 3, CD3, are striking on the protein level. They're evident transcriptomically, but barely detectable at the chromatin accessibility level. So we like to think of ASAP-seq and site-seq as companion assays that share the same modality, protein, and which is measured by exactly the same reagents, but contribute independently to chromatin accessibility and RNA respectively. And those are suitable for downstream data integration, just like what Rahul just mentioned. Also, in the interest of time, I can only show a small vignette here of how ASAP-seq was used in a model of T-cell perturbation. And I urge you to read the preprint if you need more information about that. Kelvin here stimulated different T-cell perturbations and used combinatorial hashing as a surrogate for guide RNA identities. Cells were subsequently pooled, stained with the total seq-A panel, and processed with ASAP-seq. Cells perturbed in similar pathways clustered together by chromatin, and we could also detect perturbation-induced changes in protein levels against chromatin status. And while most correlated well, there were instances of uncoupling, just like I presented before in the PBMC stimulation experiment. For example, CD69 in the CD3 and sub-70 perturbed cells, so slight increase in chromatin in gene accessibility, but there's significant decrease in surface protein levels. While the preprint was under review, the 10x multi-omic kit became available, and we explored the possibility of using our cell permeabilization conditions that were used so far with empty single cell and ASAP-seq to preserve surface antigens and input cells instead of nuclei in the multi-omic kit. This would enable simultaneous capture of 4 modalities now from the very same cells. We named this approach dogma-seq for the obvious implication that it measures gene activity across the central dogma of gene regulation, but also because it makes a pretty accurate acronym to describe our state of mind. We recognize that the poly-T mediated barcoding could be used to barcode poly-A antibody conjugates, much like we did in site-seq. And to benchmark the method, we repeated the PBMC stimulation experiment, where cells, after activation, were stained with a total seq-A panel and subsequently fixed, permeabilized, and subjected to the dogma-seq protocol. To test the performance, we compared some QC metrics using the control PBMC dataset against the 10x released nuclei multi-omic data. And we were happy to see that dogma-seq detects a comparable number of genes. It has a very good TSS enrichment, but additionally to the multiome, it gives a higher mitochondrial DNA content that can be used for lineage tracing, as well as rich surface tag libraries. For dimension reduction and clustering, with Johan's and Rahul's help, we applied the weighted nearest neighbor method, which Johan will present right after, to include now 3 modalities. And we thus recovered 25 clusters with high resolution including the activated T cell clusters. As with the paired ASAP site-seq, we could also visualize again stimulation response across the three modalities, now only captured from the same cells and not after integration. And we observed cases as seen for CD3 and 2869, where we have a heterogeneous response with changes in chromatin, RNA, and protein being uncoupled. To summarize, in this preprint and a sneak peek of the revision, we present two new multimodal tools, ASAP-seq and DogmaSeq. ASAP-seq pairs single-cell attack data with detection of cell surface and intracellular protein markers and optional capture of mitochondrial DNA. I urge you to read these preprints also in the same area with the same similar goals. Phage attack is using nanobody displaying phages for simultaneous single-cell measurement of surface proteins and accessibility. And Icicle-seq is using a digitonin-based permeabilization and a poly-A-tailed TN5, a loaded TN5, to work with a poly-T-based barcoding system. DogmaSeq is a novel adaptation of site-seq on the Multiome kit for measuring gene activity across the central dogma, with also optional mitochondrial DNA capture. And I also urge you to read TSeq, another preprint with a very similar site-seq application on Multiome, only using digitonin permeabilized cells. And with that, I would like to thank the Technology Innovation Lab at NYGC, particularly our single-cell whisperer, Peter Smybert. This work wouldn't be possible with our fantastic collaborations spanning different continents and different time zones. I would also like to thank our funding sources and BioLegend for their generosity in providing reagents and advice, and you for your attention. And now I would be happy to take questions.
Thank you, Eleni, for such a wonderful talk. And yeah, please, you can type your questions on the chat box, and also if you can raise your hand, and I will call on you to unmute yourself and ask the question. I see that we have a question from Uriol Narcís. Uriol, can you unmute yourself? If not, I can ask the question. So the question is, he can. Okay, I'll ask the question and I'll see what you can. Hi.
Hi, sorry, yeah.
Go ahead.
So my question for Eleni is, can you combine the cell hashing with other protein detection since both systems work with antibodies? I thought they weren't compatible.
No, they are compatible, especially if you use a different total-seq family. It's even easier because they're going to have different PCR handles and you can make two different libraries. But even in the case that you don't have that possibility, you can use the same family. You just need to be careful about the bridge oligo that you spike in during the barcoding reaction. It has to be compatible. So there's a bridge oligo A for the total-seq family and a bridge oligo B for the total-seq B family. And in fact, the bone marrow experiment we did combines hashing with massive phenotypic panel.
Mm-hmm. Okay, thank you. Yeah, and what's the maximum of cell hashing?
As far as cell recovery?
Yeah.
I believe 14K was the maximum we tried. Mm-hmm. Again, it's not economical if you go more than 20,000 cells, because then you start having a lot of doublets that you're going to discard. And I'm not sure what, we haven't gone more than 14K to tell you whether the transposition reaction becomes compromised after you load a lot of cells.
Okay, thank you.
Sure.
I have a question myself. So you show how you could detect both surface markers and also intracellular markers. Did you also combine that with, so I mean, extracellular markers, intracellular markers, and also RNA and chromatin accessibility, so the 4 modalities together?
So yeah, that's a very interesting application that we will be exploring in the future. The issue now is that with the multi-omic kit, because of the way protein capture is performed, it's using the poly-T barcoded oligos. So we have to use the poly-A tagged, the poly-A conjugates. It is not possible to use total-seq P conjugates, and there is some. We have some preliminary data showing that intracellular staining with total-seq A antibodies does not work very well. So it's a technical issue. We are exploring it, and yeah, you raise a very interesting point. It's not yet feasible, but I believe it will be in the future.
Thank you.